All right, guys, welcome back to Underground Science. In this video, we'll be talking about Toll-like receptor 3, specifically how HSV1 can stimulate Toll-like receptor 3 and what happens afterwards. Okay, so let's go and get started. So we know Toll-like receptor 3 responds to um, double-stranded RNA, right? As we've been talking about in our previous videos regarding Toll-like receptor 3. But HSV1, right, the herpes simplex virus 1, is a DNA virus. So then we have to see, okay, how does a DNA virus stimulate a toll-like receptor 3 if toll-like receptor 3 molecules respond to double-stranded RNA, not DNA? Okay, so let's say that we have this HSV1 virus. It enters the oral mucosa, right, of any of a person, and then from there, from the oral mucosa, what can happen is it can actually travel up to the trigeminal nerve. All right. But once it's in your trigeminal nerve or your trigeminal ganglia, it can stay latent. Okay. So it just, it's not gonna be doing much, is what that means. Okay. So let's say we have our HSV1 virus that's in your trigeminal ganglia, and it's actually latent here. It's in its latent phase. Okay. So right now, nothing is happening. Nothing much is happening. Okay. It's just basically inside your trigeminal ganglia, and what's actually happening is actually waiting to get reactivated. And that if that happens, then that's where, the damage can start. So if it's latent in your trigeminal ganglia, right, this HSV1 virus, if a certain stressor occurs, right, that can lead to the reactivation of this virus. Okay, so that can lead to the reactivation of this virus, and this happens during some stressor event. Okay, so if a certain stressor occurs, it can reactivate this HSV1 virus, which right now is latent, it's dormant. Okay, it's not doing much. It's inside the trigeminal ganglia in the latent phase. A stressor occurs, now this virus gets reactivated, and through the trigeminal nerve, it can spread to the temporal lobe. Okay, so what happens is through the trigeminal nerve, it can spread to the temporal lobe of the brain. Okay, so it's going to be our cranial nerve 5. All right, so through the trigeminal nerve, the HSV virus, right, HSV1 spreads and hits the temporal lobe neurons. Now, once that happens, right, so once it enters the neurons of the temporal lobe, okay, so let's say we represent a neuron of the temporal lobe with a large box like this. Okay, we're going to label this. This is going to be a neuron of the temporal lobe. Okay, let's write that a little better. Temporal lobe. So let's draw the HSV virus like this. Okay, so this is going to be our HSV virus. It got reactivated. Then from the trigeminal nerve, it traveled all the way up to the temporal lobe and entered a neuron of the temporal lobe. Okay, now what happens is, so this viral nucleocapsid, right, this HSV1, it has what? It has its double-stranded DNA inside of it, right? Because we said it's a DNA virus. It actually goes into the nucleus of the neuron, right? Into the nucleus of the neuron and releases its genetic material. So it goes into the nucleus of the neuron and releases its genetic material, which is its double-stranded DNA. Okay, but the thing is, unlike retroviruses like HIV, for example, this double-stranded DNA does not get incorporated into the host genome. It actually stays as an episome. Okay, so a circular double-stranded DNA molecule. And the reason why this is bad is because we have RNA polymerases, right, in our nucleus. So our RNA polymerases, right, so we'll just say RNA polymerases, can actually transcribe this double-stranded DNA. And in that process, right, double-stranded RNA can be made. And we know that toll-like receptor 3 responds to double-stranded RNA. So this double-stranded RNA could get degraded, or we know autophagy can occur, and we can form an autophagosome around this double-stranded RNA. And then we know what? This autophagosome can fuse with endosomes, right? So if we had an endosome here and we fuse this autophagosome, with this endosome, then that means that this double-stranded RNA can get into the endosome. And what's present inside endosomes, right? As we already know, toll-like receptor 3, okay? So now this is dangerous because once this double-stranded RNA, which again came from the double-stranded DNA of the HSV-1 virus because the double-stranded DNA got transcribed by our host RNA polymerases inside our nucleus. And in that process, we made double-stranded RNA. Okay, so that's how we have double-stranded RNA here. Now, double-stranded RNA can bind on the toll-like receptor 3 molecules, and then now we know the process, right? This is what we've been covering in our previous videos. Two toll-like receptor 3 molecules can dimerize, okay? And then that leads to the activation of toll-like receptor 3. That can further activate TRIF, not MyD88, right? Like the other toll-like receptors that we've been talking about, but TRIF. Okay, TRIF gets activated. Then TRIF can activate two different things. One thing it can activate is interferon beta. Why is this good? Like we said in our previous videos, interferon beta can put you at an antiviral state. Okay, so let's go and write that better. Antiviral state. So what this means is it can upregulate. So one of the things it can do is it upregulates interferon beta, upregulates MHC1 complexes. Okay, remember MHC1 complexes are on every nucleated cell of the body. And one thing it can do is it can present viral proteins. Okay, if our cells are affected with the virus, for example, these MHC1 complexes can present viral particles or viral proteins. So then cytotoxic T cells, or also CD8 positive T cells, right, can come in and destroy that cell. So of course this is good to contain the virus, but imagine if this is happening in neurons of your temporal lobe. If you have a lot of upregulation of MHC1, you're going to get a lot of CD8 positive T cells that are destroying your neurons. Okay, so that can't end up good, right? That can't be a good thing in the long run. Another thing TRIF can activate is nuclear factor kappa B. And we know this leads to a ton of cytokine production, right? So it can lead to interleukin 1 production, can lead to interleukin 6 production, interleukin 8, interleukin 12, right? And so on. Even TNF alpha. And all of this we know, that's not an alpha. All of this we know can contribute to the overall process of inflammation. It can attract white blood cells, right? It can vasodilate your blood vessels. So here, one important thing to know is so all of these cytokines, right? Not only will they damage your neurons, and in this case we're talking about your temporal lobe, what it can also do is it can start to damage your endothelial cells. Damage endothelial cells. Okay, we know that's bad because if it's damaging endothelial cells, like certain blood vessels, right, in your temporal lobe, that won't be good because then blood can leak out. Okay, that's actually what happens in HSV1 is later on, if it gets severe, you can lead to hemorrhagic necrosis of your temporal lobe. Okay, so all these cytokines, right, will lead to damage of the neurons. It can damage endothelial cells, and we know that cytokines can increase vasodilation and permeability, right? As part of the inflammatory response. One of these places is the blood-brain barrier. So what actually happens is you make the blood-brain barrier more permeable due to this excessive release of cytokines. And if you make the blood-brain barrier more permeable, that means more white blood cells can come to your temporal lobe, where all this inflammation is happening, right? Like what? For example, neutrophils, right? So if imagine this, more neutrophils come in, what do? They release enzymes and reactive oxygen species, right, to try to kill the virus off. But in the process, you damage more neurons doing this, right? You damage more neurons in the temporal lobe, right, by doing this. So as we can see, this can lead to the destruction of neurons, right? Due to all this inflammation, and all these cytokines and inflammation, reactive oxygen species, and enzymes can damage your endothelial lining. And if you damage your endothelial lining, this can increase the risk of micro necrosis, right? So what that means is the blood vessel lining, right, could become leaky, which can lead to microhemorrhages, right? So, my apologies, not micronecrosis, microhemorrhages, okay? It can lead to hemorrhagic necrosis, right, due to all this inflammation, and the blood vessels can obviously we can have microhemorrhages, which can lead to ischemia, of course, and capillary rupture, and then hemorrhagic necrosis of the temporal lobe. But it can lead to overall necrosis, okay, not micronecrosis. That's not a thing. Microhemorrhages. All right. So all this inflammation again can make your blood vessels leaky, and that can lead to microhemorrhages. Okay, microhemorrhages. And why is this bad? Because of course your brain needs oxygen, right? So if you get microhemorrhages, that's going to disrupt the flow of blood to your neurons. Okay, so you don't want hypox. That's going to further contribute to hypoxia, which will further contribute to your neurons being injured. Okay. And of course, like what important parts are in your temporal lobe? Your limbic system's in your temporal lobe, right? Your amygdala, your hippocampus. So of course you may see personality changes, hyperorality. Obviously, you could see due to excessive temporal lobe damage like this, you may see altered mental status, seizures, right? Things like that. Something interesting to know is that if due to hemorrhagic necrosis, you're going to have blood leaking, right, out of your blood vessels. So that means that you can, if you tested the CSF, you can find red blood cells in your CSF. Okay, since we're having hemorrhage of blood vessels that are inside your brain. Okay, you can see increase in red blood cells in your CSF. Of course, you can see increase in white blood cells because of all the inflammation and cytokine release. You're attracting a lot of lymphocytes and white blood cells into your brain, right? Because the blood-brain barrier, as we mentioned before, is really permeable. So you can see a lot of red blood cells, white blood cells in your CSF due to this necrosis, hemorrhagic necrosis of your temporal lobe, right? And then the another main thing to know is that you would see normal glucose levels, right? Because remember, if there's a bacterial infection, right, then you would see low glucose levels in your CSF, right, for the most part. Since this is a virus, herpes simplex 1 virus, right? Herpes simplex virus type 1, you would see normal glucose levels, right? And you could also see elevated protein levels, but the main things to know is you can see elevated red blood cells, elevated white blood cells, and normal glucose levels. All right, and so a treatment that you could consider is acyclovir because it's an antiviral. All right, so that's basically the main process of how toll-like receptor 3 is related to HSV1. Okay, and how HSV1 can stimulate toll-like receptor 3, and we know the outcome of that. All this inflammation, right, again, will lead to destruction of your endothelial cells, can lead to the destruction of your neurons, can increase vasodilation, blood-brain barrier permeability, attract more white blood cells, which can further damage your neurons in your temporal lobe, right? And then that can lead to, of course, microhemorrhages, necrosis, and then if you want, you can test the CSF for these things as we discussed. Okay, so that's the basics of toll-like receptor 3 activation and how HSV1 does.